we really create a new brand of species in a laboratory? To find out more, stay tuned to this lesson brought to you by the Division of Education, Innovation and Energy e-learning program. I am your beloved biology tutor, Lenetta Cowie Clarke. And today we'll be focusing on genetic engineering. So, to continue with this topic, you should know about natural and artificial selection, genetic variation, and speciation. Our objectives today are to get you to be able to define genetic engineering, describe how genetic engineering can be used to change the traits of an organism, and discuss the possible advantages and disadvantages of genetic engineering. What do you get when you cross a lion and a tiger? We have a liger. And here we have two liger pups. Is that natural? I'll let you think about that for a minute. These are the famous glowfish. Bioluminescence fish engineered to glow in the dark. And here is Ruffy. Ruffy is a cute puppy that also glows in the dark. So here's a picture of Ruffy in the night, and Ruffy's paw is also glowing in the dark. So what is genetic engineering? Genetic engineering is also known as genetic modification. And genetic engineering is the use of technology to alter the genetic Genetic engineering is the use of technology to alter the genetic makeup of an organism. What happened? This is direct gene manipulation in a laboratory setting. How is it done? Now, according to the National Human Genome Research Institution, this is achieved by using recombinant DNA or rDNA, or DNA that has been isolated from two or more different organisms and incorporating that into a single molecule. So basically, we are taking a trait that we want, for example, bioluminescence, and slicing it or splicing it into the DNA or RNA of another organism, practically changing its phenotype, changing what we actually see. In the past, diabetics depended upon insulin derived from animals. Now, thanks to genetic engineering, human insulin is mass-produced in the laboratory, and this is how it is done. They take a segment of DNA from a non-diabetic person. So for example, here is a segment of DNA, and the part that they're interested in is this piece right here. They will remove or isolate that piece of DNA. Then they will take the circular DNA from a bacteria, otherwise known as a plasmid. They will cut the plasmid at a very specific location and incorporate the human segments that codes for insulin production into the plasmid DNA. So here we have the recombinant DNA that has been inserted into the bacterial plasmid. They will then insert or reinsert that recombinant DNA into a bacterial, and the bacteria will do what bacteria do best. It is going to mass produce and start producing insulin. In the lab, all they will do now is isolate the insulin from the bacteria, purify it, and package it, and it's now ready for sale. It is going to be human insulin because the gene was taken from a human cell. The gene that codes, or the segment of DNA that codes for insulin production. So all we're basically using the bacteria for is as a host to mass produce the insulin. Another example is the use of genetic engineering to produce the hepatitis B vaccine. Similar to insulin production, where they will remove a segment, the antigen producing segment of the gene from the hepatitis B virus, and they will insert that again into the plasmid of a bacterium. So here we have the inserted segment. However, instead of placing this back into a bacteria, they will then place this whole recombinant DNA into a yeast cell. And the yeast cell will then do what yeast cell does. It mass produces and reproduces, and it will ferment into a tank, and the end result is mass production of hepatitis B antigens. They will then extract that, purify it, and create your hepatitis vaccine, all done in the laboratory. In terms of foods, we have lots of genetically engineered foods which we eat. So we have squash, zucchini, pawpaw from Hawaii, cotton, corn. We all know about 6-weeks corn, which is a genetically modified version of the variant of the corn that we used to use long time. Soy is a genetically modified foods, canola, sugar beet, alfalfa, just to name a few. So what are the advantages and disadvantages of genetic engineering? So genetic engineering has many, many benefits. They are used for DNA tests, gene therapy, genetic fingerprinting, so crime fighting initiatives, disease and pest resistant crops. So they can use genetic engineering to create crops that are resistant to different kinds of pests or disease, and therefore they would of course last longer and you have a better production. You have fruits and vegetables which last longer on the shelves. For example, we have tomatoes which look brighter, which are bigger and probably even tastier, and they would last longer, so they would survive the travel or the transportation from farm to market. Of course, they have used genetic engineering to incorporate minerals, for example, keratin into rice, which are grown in areas where night blindness is prominent. We also have another benefit of genetic engineering is that it is cost effective when creating vaccines and other medicines. And because a specific gene was removed and inserted, then we only have that desired characteristic being expressed in the organism that we want. And of course, it is faster than selective breeding, a topic we did before, and we know that selective breeding takes a very long time. So genetic engineering is much, much faster than selective breeding. However, because it is labor intensive and quite a very specific, why is the word gone for me? It is expensive because it is labor intensive, and we have the potential of having some negative environmental impacts due to the insertion or the creation of these new organisms with these recombinant genes. So because the technology is fairly new, we don't know what some of these long-term effects may be. Some of them may create cancers, we don't know. the data is not there to give us enough information about what some of the effects of these recombinant or these GMO products, for example, the foods that we eat, are going to have on our systems. And of course, there are some social impacts. Because it's an expensive project, who will benefit from such genetic engineering projects? Would it be for everyone or just those who can afford it? And naturally, we have ethical issues being raised. Is it going to be regulated? How far can we go with genetic engineering? So for example, these are two salmons. The one in front is a natural or the normal salmon, and the one in the back is a genetically engineered salmon, and they're both of the same age. So what we will notice is, obviously, if you are a fisherman, you are going to bring in or make more profit with the larger salmon. But do we know what the effects of eating meat that has been genetically engineered will have on our system? So we have to weigh or balance the profits or the advantages against the disadvantages. We have cows that have been genetically engineered to generate milk that is very, very similar to human milk. Of course, we can see the benefits in that, but there are also some other ethical issues in terms of how far are we going to go with genetic engineering. Here we have a craze in China where they have been creating pigs that are small enough to fit into a teacup, and these are used as pets. So the question is asked, how far can we go, or how far is too far? Can we create designer babies where we would want to dictate what is expressed? If we can eliminate different diseases, for example, Down syndrome, sickle cell anemia, if we can remove those corrupt pieces of DNA from an organism and create new species or create organisms or babies that do not have these diseases, would we be willing to go that far? And who is going to benefit from such designer babies? How far is too far? I would like you to think about some of these implications, read up about them on your own, and I do thank you for joining us for this segment of the Division of Education, Innovation and Energy e-learning program. That brings us to an end to this segment of biology. Thank you and stay safe.